Welcome back to the class. We are going to continue after the apomixis polyembryony. What is called embryony, children? It is nothing but formation or development of embryo in a seed. Normally one seed contain one embryo is usual, but occurrence of more than one embryo in a seed is called as polyembryony. Observe this picture. Polyembryony, embryo sac of Ulmus glabra, zygotic and antipodal embryo. One egg fuses with sperm and produce zygote. Zygote development embryo is normal. So in the zygotic embryo and antipodal embryo, both you can see in this embryo sac. Hence it is called polyembryony. Okay, children. Now let us continue the next one, polyembryony. Occurrence of more than one embryo in a seed is called polyembryony. Polyembryony was first reported in oranges by Anton von Leeuwenhoek in 1790. Polyembryony is divided into several categories. 4 categories based on its origin we are going to see one by one. So polyembryony is divided into 4 categories based on the origin where it is developed. The first one, cleavage embryony. Example, orchids. Pronounce it as orchids. Second one, formation of embryo by cells of embryo sac other than egg. Very simply you can understand. Formation of embryo by cells of embryo sac other than the egg. Other than the egg in the embryo sac, what are they? Synergids are there. Antipodal cells are there, not endosperm. Synergids and the antipodal cells from which the embryo is developed. Hence it is called a what? Polyembryony, because it is not developed from the egg. Okay, so synergids, example, Aristolochia bracteatum. Refer the pollination in Aristolochia. Picture is available. Next, in this synergids of Aristolochia, more than one embryo is formed. Next, antipodals of Ulmus. Endosperm from Balanopora. This is rare case. Endosperm from Balanopora. So polyembryony takes place from embryo sac other than the egg. Synergids, from synergids Aristolochia, from antipodus Ulmus, from endosperm Balanopora. Now we are going to look at the third type, development of more than one embryo sac in the same ovule. How it is possible? There are two possible. Sometimes out of 4 megaspores cells, instead of one, more than one will start to develop. We studied monosporic embryo sac, bisporic embryo sac and tetrasporic embryo sac. The same case. So more than one embryo sac within the same ovule will lead to the polyembryonic condition. So the embryo sac that derived from the megaspore mother cell, that is called MMC, where more than 2 megaspore mother cells are used to develop more than one embryo sac, is called polyembryony. Keep remember, 1 megaspore mother cell which divide meiosis, by which gives 4 megaspore cells. Out of 4, usually 3 will disappear, 1 will grow as one embryo sac, is called monosporic embryo. Sometimes 2 megaspore cell or more than 2 megaspore cell which develops in the ovule as embryo sac will give rise to polyembryony. Example, derivatives are 2 or more megaspore mother cells. Example, casuarina. Next, activation of some sporophytic cells of the ovule. You can see the sporophytic cells such as nucellus or sometimes integuments. Example, citrus and CGM. So other than the embryo sac cells, sporophytic cells in the ovule called nucellus or integuments which gives rise to polyembryony. Example, citrus. So what is the significance of this polyembryony? You can see the polyembryony condition in this ulmus. Antipodal embryo is seen and the zygotic embryo is seen. Zygotic embryo is normal, but more than one embryo is seen in this seed where zygotic embryo and antipodal embryo, we call it is polyembryony condition. So polyembryony, what is the practical application? Polyembryony, seedlings formed from the nucellar tissue in citrus are found better clones for orchards. People are cultivating fruit yielding plants or trees called orchards, we say. Embryos derived through the polyembryony are free from the viral infection. So we can use for plant tissue culture also. So embryos derived from the polyembryony are found in virus free. Okay children, so now polyembryony means what? More than one embryo that found in a seed is called polyembryony. 4 categories are there, namely cleavage polyembryony observed in orchids, formation of embryo by cells of embryo sac other than egg such as synergids, antipodals and endosperm, development of more than one embryo sac within the same ovule, that is one reason, activation of some sporophytic cells of the ovule which gives rise to polyembryony. Welcome back to the class. We are going to look after the last part in the lesson, that is Parthenocarpy. 10th standard itself you might have studied this term Parthenocarpy. Simple definition: development of fruit without act of fertilization is called Parthenocarpy. In markets and all, sometimes when you go and buy fruits, seller used to say seedless fruits, grapes, papaya, apple, sometimes tomato also we can see. These fruits, if you observe, inside there is no seeds. So how, why seeds are not produced? Because these fruits are developed from the ovary where no fertilization occurred. So without act of fertilization, ovary directly develops into fruit called what, children? Parthenocarpy. Okay, let us see the definition. Parthenocarpy: fruit-like structures may develop from the ovary without act of fertilization is called parthenocarpy. The fruits developed by parthenocarpy are called parthenocarpic fruits. Sometimes we used to call seedless fruits. These parthenocarpic fruits will not have any seeds, no seeds. Many fruits are produced commercially by orchards where the fruits in which seeds are absent. We called simply them as seedless fruits. They develop through by the parthenocarpic technique. So many commercial fruits are made seedless. Hence they are called seedless fruits. Example, papaya, grapes and banana. Many is there. In 1963, Nietzsche, who classified the Parthenocarpy in plants into different types. Three types we are going to see one by one. What are they? The first one is genetic Parthenocarpy. Second one, environmental Parthenocarpy. The third one, chemically induced Parthenocarpy. Okay, so let us start from the genetic Parthenocarpy. Parthenocarpy arises due to the hybridization or mutation that takes place in plants. When we hybridize, or sometimes naturally the mutation occurs in plants where the fruits are formed without act of fertilization is called genetic Parthenocarpy. Citrus fruits, cucurbits, that means cucumbers, are some of the examples. Environmental Parthenocarpy, the second type. Environmental Parthenocarpy in the sense the condition of the environment is not favor for the plant to go for the usual process of reproduction. So, in their flower, the ovary which directly develops into the fruit. It is called environmental Parthenocarpy to avoid certain extreme conditions, frost, high temperature, or no water in the soil, such as so many is there. Environmental conditions like frost, fog, very low temperature, or very high temperature. To escape from these environmental conditions, extreme conditions, where the fruits directly develop from the ovary in plants. It is called environmental Parthenocarpy. So when we treat some plants with low temperature, it is possible. Low temperature for 3 to 19 hours induce Parthenocarpy in pears. So pear fruit develops without seeds when they treat with very low temperature for 3 to 19 hours. Hence it is called environmental Parthenocarpy. Now move to the next children, chemically induced Parthenocarpy. Sometimes farmers used to spray some chemicals called phytohormones or plant hormones. These hormones, they naturally induce the ovary develops into fruit without act of fertilization. Hence it is called what? Induced Parthenocarpy. People used to say artificial. Commercial aspects, artificially the fruits are developed from the ovary by spraying some chemicals. Application of growth hormone like auxin, 11th standard you might have studied, indole acetic acid, phenolphthalein acetic acid. These are all some of the auxins, natural auxins. When we spray, and gibberellins, when we spray on the flowers, ovary is present in the flowers which it develops into fruit-like structure. Hence, it is called induced Parthenocarpy. So, chemically, when we use a fruit to develop from the ovary without act of fertilization. What are the advantages? What are the significance? Why we want to do? Because it increases the fruit formation in the plants. So, orchards gets more economy, more revenue they will get. Let us see. Seedless fruits have great value in horticulture. They have a very great commercial value in industries. Parthenocarpy very much useful for the preparation of jams, jellies, sausage, fruit drinks, etc. Sometimes you might drink fruit squashes where you can't see seeds. Food industry, those who are extracting those who are extracting juices from the fruits, they are preferring only seedless fruits. Next, the quality. The edible portion, high proportion of the edible part is available since seeds are abortive. Abortive means seeds won't be formed. So more edible part which can be seen in fruits. So it has more commercial value. So what is called parthenocarpy, children? The formation of or the development of ovary into fruit without act of fertilization is called parthenocarpy. Parthenocarpy was classified into many types. In 1963, NITS classified the parthenocarpy in plants into three types: genetic parthenocarpy, environmental parthenocarpy, and induced parthenocarpy, chemically induced parthenocarpy. Using auxin, gibberellin like hormones, they induces fruit formation without seeds is called as chemically induced parthenocarpy. Children, you might have understand these three topics such as apomixis, polyembryonian, and parthenocarpy. Unit reproduction in plants, asexual and sexual reproduction, chapter 1 got over. Go through the videos once again carefully. Take notes. If you don't have book, better take notes. Thank you, children. Good luck.